Hello, in this video we're going to be talking about amino acids, specifically amino acid structure. But before we dive in, let's take a big picture view. Where do amino acids fit in human metabolism? So let's think about the hemoglobin inside red blood cells. So hemoglobin picks up oxygen in the lungs and delivers it to tissues, kind of like a car picking up a passenger and dropping them off, right? So here we have O2 in the hemoglobin being carried off. And amino acids are the building blocks that make hemoglobin and basically all proteins possible. So it's important to recognize the structure for amino acids. And so the basic structure for the amino acid is going to be like this. So you're going to have the alpha carbon in the middle attached to a hydrogen and an R group, and then an amino group as well as a carboxyl group, just like this. And so here, if we were to go ahead and label, the alpha carbon would be this carbon right here. This is the R group, and this is the hydrogen attached to it. This is the amino group, and this is the carboxyl group. So the R group is the unique side chain that distinguishes the 21 different amino acids. And so every amino acid shares the same backbone, but what changes between them is this R group. One really important thing to note is that the alpha carbon is usually chiral. So a chiral carbon has 4 unique groups attached to it. And here we see that the alpha carbon is attached to a unique R group, a hydrogen, um, a amino group, and the carboxyl group. And so this is consistent throughout all amino acids. And this applies to the chirality of the alpha carbon is present in all amino acids except for glycine. Now, the structure for glycine is like this, right? So we have the hydrogen here, and I'll draw the R group here in a second, but I just want to show you that all amino acid backbone structures are exactly the same. So here, just like this, the basic amino acid structure. And what's different when compared to other amino acids is that glycine has another hydrogen here. And so that makes the alpha carbon no longer chiral. It's the presence of the other hydrogen that is bonded to, which corresponds to its R group. And the reason being, like I said, is that chirality depends on the atom, the carbon atom, having 4 different unique groups. And since there's 2 hydrogens, which are exactly the same, the compound is no longer chiral. So we talked about how amino acids, except all amino acids except for glycine seen here, are chiral. But what does that mean and why is it actually important? So chiral molecules rotate plane polarized light because their asymmetrical 3D structures cause them to interact differently with the light's electric field, slowing down one circular component more than the other. And this leads to a phase shift and rotation of the plane. And the reason this is crucial and because it allows for scientists to distinguish between enantiomers, which are mirror image molecules, which have identical physical properties but rotate light in opposite directions. And a lot of the times you might think like, okay, they're the same structure, they're organized the same, but they just rotate light differently. And you might think that, that usually doesn't make a huge difference, but a lot of times in medicine and in different drugs used, it makes a huge difference. What I mean by this is that in biology and pharmacology, there may be one enantiomer of a compound that might be a beneficial drug, while its mirror image is inactive or it could possibly even be harmful. Another way to show amino acids is using what's called a Fischer projection. So this helps you see how the four groups are arranged around the chiral alpha carbon. And so here we have the carbon in the center connected to the top here to the carboxyl group, bottom here to the R group, and then the amino group, and then of course the hydrogen. So there's two different orientations for amino acids, and that's L and D. And so to determine the orientation is you look for the amino group, right? And so let me go ahead and draw the other orientation. So you look for where the amino group is, right? Just draw it the same way, the carboxyl group at the top, the R group at the bottom. And you look to where the amino group is. If it's on the left, that orientation is going to be L. And then if the amino group is on the right, it's going to be D. And D stems from the Latin word dextra, so dexterous, as in the right. And so the reason why this matters is because the L form is the only one found in the human body. So let's go ahead and summarize all the points that I talked about in this video. So initially we talked about how amino acids, they all share the same backbone. And that backbone consists of the amino group, the carboxyl group, and the hydrogen, and then the R group as well, which is unique to each amino acid. Next, we talked about how the chirality of amino acids and how all amino acids, except for glycine, are chiral. And the reason being is that glycine has the hydrogen atom for its R group, right? And so that makes the compound non-chiral. And the last thing, we talked about the different orientations of amino acids, the L and D, where the amino group is on the left and right, respectively, and how the L-amino acid orientation is the only one found in the human body, while the D isn't. The D amino acid orientation isn't. And so that's going to be it for this video. I hope you learned something, and stay tuned for the next one. Thank you.